showing up, everybody, for, what meeting is this? The 1236 of the Entomological Society of Washington. There's a storm raging outside, so we really appreciate anyone who could come in person or virtually. We've enjoyed some lovely pizza. Let's hand it over for our minutes.
Hi, I'm Gary Bevel,
Sorry about that.
recording secretary for the society. And I usually read the minutes of the last meeting, and that's what I'm going to do now.
Okay, scooch to your.
Pardon?
So the camera can see you. Yeah.
There you go.
Oh my God. The 1235th regular meeting of the Entomological Society of Washington. Let's see. Was called to order at 7 p.m. by President Matt Buffington. As usual, the meeting took place at room WG 33 in the National Museum of Natural History. In attendance were 14 members and 2 guests. Virtual visitors numbered 21 for a total of 37. The minutes of the February meeting were read by recording secretary Gary Bevel and were approved. President Buffington noted that the executive committee recently discussed the need for an update of the bylaws. A vote will occur on these changes and other business of the society at a future date. President-elect John Weber offered an update regarding the society's annual banquet. This event is now scheduled for June the 15th and will be in the evening. Dave Wagner will be the invited speaker. The venues are being evaluated at this time. Society members are encouraged to advise Don regarding their preference of potluck or catered services. Email is fine. Program Chair Alan Norbaum introduced the speaker of the evening, Andrew Jensen of the Systematic Entomology Lab in Nelsville, who presented insect cuticle is for entomologists, not engineers. Dr. Jensen began by noting that cuticle research has been done mostly by engineers, not entomologists. There are 3 parts in cuticles: the epicuticle, the exocuticle, and the endocuticle. Research has shown great strength in weevils, especially the can't read it. Snouts. Eric Loader in 1992 found that there are zones of strength in different parts of insects. Earlier studies were confusing, lacking information of what insect zones were being stained. Experimental consistency is strongly needed in cuticle research, and these opportunities for comparative analyses between and among entomological taxa. Anything that sounds right or wrong?
Yeah, but motion to approve the minutes. Thank you. So moved. Thank you for that. So I'm going to introduce our speaker tonight. That's Jonah Ulmer. So quick introduction here. He did his undergrad in Penn State, working with Istvan Miko on Palearctic Seraphonidae. After that, he worked in Stuttgart, Germany, doing his master's under Lars Krogman at the State Museum of Natural History, Stuttgart, and University of Hohenheim on the taxonomy and exochronology of Seraphonidae. He continued working on his PhD with Krogman at the museum working on a ground plan with reconstruction and the evolution of early Chalcidoidea, along with working on the evolution of exocrine glands in Hymenoptera, examining the exaptation of glands from non-social insects to social insects. He is currently a USDA-ARS postdoc working on Diprionidae, conifer sawflies, and evolution with Matt Buffington and Dave Smith. And we're happy to have him.
All right. Thanks everyone for being here. In a crowd tonight. All my friends and everyone out in Zoom land for not getting wet and breaking the sword. Yeah, so I would like to talk today about the captive audience about exocrine glands in Hymenoptera, which is a niche within a niche that I've been really interested in for many years. I'm always looking for unwitting suspects to upload it all onto. The first thing I want to get out of the way, I'm not a chemical ecologist. I'm a morphologist, taxonomist, and I bring that up now because I've often been asked about chemical ecology and exocrine glands, and I'm not the one to ask. But exocrine glands outside of chemical ecology and social biology present really unique slew of evolutionary questions. Both in a taxonomic sense as a character complex that can be used, and also in understanding the broader strokes of the evolution groups via the biosynthetic pathways. And it's important to kind of understand in exocrine glands that you have these two sort of parallel evolutionary lines that are occurring. The morphological evolution of a gland, such as you see here, these are the tergal scent glands of pentatomids, right? And there's variation within, this is a subfamily that can be used as a diagnostic character for taxonomy. And then the other line is the biosynthetic pathway. So the other little cladogram here shows the broad strokes of Hymenoptera with variation in the volatiles in the force gland. And the way that the biosynthetic pathways shift as you go from phytophagy to parasitoidism to brood care, social behavior, and back to phytophagy. And so I think before I start, it's really important to understand glands are a really plastic suite of organs. They evolve and adapt incredibly fast, and especially in insects. Insects are a great sort of case study for exocrine gland research, primarily due to some inherent features of insects and arthropods in general. So insects are a tube. They are a single epithelial layer that is just kind of being folded onto itself. They're basically organic origami. And what you get, right, is you get cuticular deposition that gets thicker or thinner, sclerite, conjunctiva. For more thorough analysis of this, I recommend you watching your Jansen's talk this week. But this cuticular deposition, right, is important due to the fact that all epithelial cells in insects are inherently gland cells because they are producing cuticle, they're excreting it. And so the sort of infrastructure of producing an extract within a cell is already there, right? The organelle ratios are right. So it's just a very quick shift of what you're producing, and you have an exocrine gland. And a great example of this was actually not in my talk originally. I was having coffee on Monday, and I heard an NPR story. And I was sitting in another room, and NPR goes, "Oh, we're talking today with an entomologist about exocrine glands." Ran and turned the radio up. It's my time, finally. And they were talking with Kobe Shaw at University of Kansas. He's a chemical ecologist, but they were talking about exocrine glands in German cockroaches. And so what you see here is a male and a female German cockroach. And the female is kind of nibbling at the butt of the male. And so in the courtship of German cockroaches, the male, from a gland on their abdomen, produces this tacky sort of secretion that's mostly made of maltose. And the female is like this, it's a nuptial gift. So she kind of climbs up onto his back and starts nibbling away at this excretion. And her saliva breaks it down into glucose. And cockroaches like sweet things. And so while she's in the process of munching away at this extract, he has time to copulate. Pest agents also are aware that cockroaches like glucose. And so often poisons used to deter cockroaches are spiked with glucose. And female cockroaches in recent years have gained an aversion to sugar. And so what was happening was the female cockroach would climb up onto the male's back and start eating away at it. They would break the maltose down into glucose, panic, and have an aversion to it and run off so the male could eat. And so males, over the, this is over the span of a study, right, over a human lifetime, the males have shifted the composition of their extract of nuptial gift from maltose to maltotriose, which is much harder to break down into glucose. And so it gives them more time to mate, or females working away at it before she notices, oh, it's glucose, it comes off. So that's, that's within the last 50 years, this transition has been reported. And so this just goes to show egg-stream glands evolved insanely fast in insects. And it's a great system for looking at, within your lifetime, adaptive radiation, and looking at the homologies and convergencies in some of the structures. So in the first slide, I mentioned sort of two halves of egg-stream gland research. One is the taxonomic utility, and the other is asking some more interesting evolutionary questions about function and convergence. So I will start with the taxonomic utility. So I, as I mentioned, started my academic career working on Seraphonidae, which are a very small group physically and diversity-wise of parasitoid wasps. And our lab's project was working on developing a character system for delimiting genera and species. And so while this was the overarching project, I was given this very small project to work on this gland, which is a familial synapomorphy of Seraphonidae. So this is a character that sort of defines the group in some way to allow it to be separated from others. And it's this rather large gland, relatively speaking, on the abdomen of Seraphonids. And you can see here that it is completely warped, basically, the shape of the tergite. The tergolapidae, so where the muscles attach, have been shifted inwards from the lateral edges. And it's basically completely altered the biomechanics of their abdomen to have this gland there. And so it obviously must have had some significance. And so this gland was studied previously by a scientist called Sartre. He was probably the last major Seraphonidae taxonomist. And in the 90s, he wrote a paper discussing this gland and said, well, I know it's a family level character. It might be a generic character, but I'll check that later. He died probably a year after that paper, so he didn't get around to it. So it was sort of passed down to me. So a very quick introduction to Seraphonidae. They have been called, and I'm directly quoting, mind-numbingly morphologically monotonous. Someone said that to me, Adam, say. So I've kind of cherry-picked here some of the diversity. So these are a lot of the different genera. The vast majority of the genera are monotypic. Vast majority of species are in 1 of 2 genera, Aphtogmus, Seraphon. We know almost nothing of their biology. What we do know is that they are extreme generalists, and most of them are hyperparasitoids, which makes them incredibly difficult to right, okay. So back to my gland. Now I can give you a brief history of the water supply, or evaporatorium, as I've been calling it. So like any good first year research student, I quickly went and I found hundreds and hundreds of papers on the topic, and I read probably a tenth of them, and then the rest just kind of sat in Google Drive forever. But what I did find was, like all good scientific discoveries, it was not attributed to the right person. So Nies in 1844 refers to notice the gland, and mention it in one of his descriptions. And then Bruce in 1902 was the first person to actually attribute a function to this structure, formally kind of describe it. And I will tell you what Bruce's functional hypothesis was. I need to preface it by saying it is possibly the most insane functional hypothesis I've ever heard about a structure. It requires a complete breakdown of evolution and human consciousness and our basic understanding of the universe. Really grasp, right? So Bruce had a phase at the start of the 20th century where he would go around Texas and basically excavate whole ant nests and just dump the dirt and then sift through it and describe everything in that pile. And he described a lot of interesting equal lines of ant nests. And one such taxa he described was Acetiniades subactris, which is this little guy here. And so he saw this, said this is an equal line in acetone army ant nests. Since, over the last 120 years, no one has been able to find this anywhere near army ants. It's very likely they just dragged the body into the nest and it's not there. Or it just got kind of picked up with the rest of the dirt. And so he noticed this gland, or this, he didn't know it was a gland, this sort of reticulated patch on the abdomen. He said, well, I have seen these acetone army ants, which are functionally blind. I've seen them kind of walking around and antenating various other ants and these equal lines. And occasionally, when they antenate an equal line, they attack it. So he said, this patch is basically a rail that the ants read to know that this is an intruder. And so we're gonna break that down to discuss how strange of a world it would be if everything evolved to make it easier for predators, right? And then putting that aside, we can think about the fact that this entire hypothesis hinges on the fact that ants have a tactile-based writing system and are literate, which I'm sure there's probably a few fringe sociobiologists out there who would make that argument, but I'm not one of them. To Bruce's credit, though, this hypothesis came out a few years before cuticular hydrocarbons were discovered. So he was on the right track, just not in the right train, basically. So, I get where he was coming from, still crazy. And so then getting back to this, Waterston then, said, oh, I think this is a respiratory function. Oglobin looked at it, a different group, and said, it's no trachea, I think it's a gland. And then Dessart, who I mentioned, he looked at it, said it's a gland, and now we're at present day. So this is kind of going in tandem with the overarching project of this grant, which was finding a robust character complex for discriminating genera and species within Seraphon. I don't know if I mentioned Seraphronids at the time, males could be separated to genus with relative confidence based on the shape of the antennae. Females, even to genus, was basically a coin flip. There were some shape differences in the thorax you could use, but it was very shaky characters. Species were completely out, right? Totally dubious, all species concepts. So we were working on this for a while, and we found that male genitalia is a really robust character complex that separates genera really nicely. So a lot of characters, species, it separates really nicely. But of course, the main problem with male genitalia as your primary character is it only works on males. And for those of you who have never seen a confocal laser scanning image before, wasp genitals don't naturally glow green. Well, I guess they do naturally shoot a very high-powered laser at them, right? They autofluoresce. So yes, they do naturally glow green. Okay, so we get back to it. So we have a great character complex for separating males, but the problem still lies, how do we separate females in genus? And so, you know, I was doing this gland project, dissecting stuff. So I took, this is Seraphron and Aphanogmus, and I cut out the gland, and I said, well, they're pretty distinct. You know, you have some variance in the reticulate structure. You have some variance in the curval apidames, right? How the sclerites form. This acrotergite's different. And I give credit to Oglobelin and Waterston. They took no artistic liberties back in the 20s and 40s, you know, with their little microscopes. They did a really good job of defining these. I just think no one ever cared enough to put two and two together. Okay, so then we now have a character to put females confidently into a genus. But what about species? So again, we looked at genitalia between two species. This is Aphanogmus fumipenis, Fasciopenis. These were species we're pretty confident about. There's the genitals, and there's the genitals of all the other species in their complexes. So again, not super confident. But again, I looked at the Waterstones Laboratorium, those, you know, two original ones, and go, oh, it's also different between them. And they're both, if you squint, Aphanogmus like, right? They have this relatively straight, well-defined inner margin spherization of the apidames, very small evaporatorium surface. So it seemed like there was maybe some species level variance in the glands as well. And so I just wholeheartely dove into this. And over the course of a few years, I have dissected several thousand Aphanogmus and Seraphron abdomens. And it actually is what got me my master's and PhD, because the only curator on earth who would willingly send me bulk specimens, knowing full well I would just destroy them and never send them back, was Lars Krogman. So I said, all right, you know, he was there from the beginning. And so I looked, you know, at species level variance. And so this is 4 different species of Seraphron based on male genitalia. We have 4 distinctly Seraphron-esque Waterston's evaporatoriums, but each one, I believe, there's enough characters that kind of separate species based on this. This is intraspecific variation within one species of Seraphron based on male genitalia. Again, there's some slight variance in the reticulate network, but the intraspecific variance is pretty low. So this was supposed to like come in and be a big surprise, but it didn't. So the last thing we wanted to do was, can we match males? Because that's the only thing the genitalia can't do. And this is sort of a paradox, right? If you're trying to find a character to identify females to species and match them to species, how do you find the females to check that for to begin with? And so I used a monotypic genus that we're pretty confident in. And I got the male and female, and they match. The males have this a slight dark patch at the base. Not sure why. It's also in this other one, right? You see the male has a darker patch, the female doesn't. But so we can now match males and females on the species level. So genitalia, we use as a robust character set, and we use Waterson's evaporatorium. And we also tested this with CO1 barcoding, and these are indeed the same species. So it does work. So then I started looking at all this little monotypic genera, right? I looked at Acetinides, right? Bruce's species. That's that one. Eliza Seraphron, Synarsis, some other one, I don't remember. Just meat crops. Right? I kept going and going, and they were all quite distinct. This weird little gland on the back of a weird little wasp, a species level fingerprint to identify that. And it was a very long and arduous project. I will say after I published this, probably 8-9 months later, my best friend, who's a molecular biologist, worked out the primers in exactly the right, thermocycling agents to just do very efficient and rapid barcoding. So this, and I begged her just to cite this. It'll be the only citation I ever get. But yeah, I mean, this just shows the taxonomic utility of just the morphology of an exoplanet, right? And I will get ahead of the questions at the end. I have no idea what it does. And part of the reason, right, is this is truly a novel structure. I've looked at countless Hymenoptera families, subfamilies, genera. There's not any other ones that are here like this. And novelty is really cool when you're publishing a paper, right? Everyone wants their title, novel structure, novel this. It is completely evolutionarily worthless to be novel, right? You're an island, you have nowhere to go, you have nothing to compare against, right? To feed off of. And this is a truly novel structure. Great for taxonomy, not great for evolutionary questions. And so this leads to the next project in the next gland. And so, yeah.
Can you move the little video
Can you-
thing?
Oh,
Can you
yeah.
hit the little blue button for whoever's in the waiting room too?
Okay.
Thank you.
Sorry.
I don't know how that got past you. That's why that.
Okay, cool. Sorry, Becca. All right, so
It looks like a cool picture.
it is. So I mentioned at the beginning, right? You have taxonomy as a functional use of exocrine glands and insects. The other is ask some really interesting questions. And so this next one I'm going to talk about is one that this paper was just published Sunday after a very long time. And this was my COVID project, right? I was in quarantine, museums were closed, I couldn't get specimens, I had all of these images, this data, I just worked on this. And it's ironic you'll hear throughout this because this gland is really important for social immunity and making sure you don't get sick in groups. And so I'm going to talk about pellicidity, which is this wasp. It is an even smaller group than Seraphonidae, although much more charismatic. They are parasitoids of Phylophaga, so May beetle larvae underground. They use their other long tail to kind of fish for the soil. They're an exclusively New World family. The most common species is Pellicinus polyturator. It has a very unusual sort of novelty about it in that it is geographically partitioned parthenogenesis. So north of the equator, no males. It's just females. They do just fine by themselves. South of the equator, you get males. And then they just, go about it the whole way. Is this not? Oh, there we go. Okay. So there was a Hymenoptera anatomy project that occurred 15 years ago. And during that, they were examining the thorax of pretty much all Hymenoptera. And they noticed this depression in the metapleuron of pelicids. Now, this can be easily kind of passed off as being the metacoxal particularly sort of depression. But there were little pores inside it. And as the adage goes, where there's pores, there's glands. But this was rather novel because thoracic glands aren't super common in non-social Hymenopterans. You basically have the coxal glands and placonids, and you have the euglyoline sort of scutellar disc gland. That's about it. And I will get ahead of it because I know Brendan will bring it up at some point. I know the metapleuron is not technically part of the thorax in the plesiomorphic condition, but for the sake of this, we're going to stick to the thorax. So I decided to take a closer look because I had the time. And so I cut open some pelicinids and I noticed this huge, I can see it, huge gland, relatively speaking. And this gland is like 300 microns. It's massive. And it corresponded to that sort of depression of the pits on the metapleuron. And this gland wraps really tightly around the dilator muscle of the proteospiracle. And I was looking at this, I know this gland. I had this tingling feeling. I've seen this before. So I decided to take an even closer look. And again, I took some CLSM photos because they go over really well in talks. And I did some serial block-based SEM, also known as TEM for people afraid of microtones. And what I noticed was, this wasn't just some little tweaking of the biosynthetic pathways of epithelial cells, right? These were really, really advanced gland cells. These were class 3 gland cells, right? So I can very briefly give everyone a rundown of gland classes. Class 1 gland cells are your mom and pop salvador things, right? Just a cell gives off vacuoles until it's out of membrane and then it dies, right? Pretty basic. Class 2, they're like cryptids, right? Like some termites have them. Everyone always knows someone who knows someone who's a class 2 gland cell. Class 3 gland cells are the luxury line Cadillacs gland cells. They're really expensive to make, big, and they're just impressive because they require sort of a whole restructuring on a cellular level, an ultrastructural level. So class 3 gland cells are like nesting dolls. It's basically 3 cells, one nests inside the other, and then you get another one kind of in that. Main one is the gland cell. It produces the extract, pumps it into the second cell. Second cell sort of packages it, pumps it up in the third, which then basically deposits it out. What this allows is the gland never just sort of depreciates. It can just keep going and going and going until natural cell lysis, but it can pump. So these glands are big, they're very intensive to make. So this gland must be very important. And again, I feel like I've seen it. So I start looking at other insects. I look at close to 100 taxa across 46 families. And it's really important. Pit does not mean clan. Insects have a lot of pits. They don't all equate to a gland. So what I was examining for was some structure internally, a gland I can see wrapping around this proteospiracle dilator. And only one family had a similar structure of ants. So this is the metapleural gland here of Adacephalotes. And it's elongate. This is actually a dual chambered metapleural gland, which is glands. And so this leads me to a minor tangent on ants. It's like I recognize. So yeah. So Brennan will recognize this beautifully done 3D reconstruction. So as I mentioned when I was going through examining these families, my primary criterion was a gland wrapped around the proteospiracle muscle and spherical. and Apodane. I could not find in any ant literature from anywhere any reference to a podiospherical Apodane or muscle. I thought I was going insane. The ants are pretty well studied. Just wasn't there. Oh, hang on, we gotta let it cool in. Just wasn't there. And so I finally gave up and I contacted a collaborator and a friend of mine, Adrian Richter, who's a very famous pharmacologist. And I said, Adrian, does this muscle just not exist? What am I missing? Well, I've got some CT, micro CT data. I'll reconstruct this. And he did. This is one of the reconstructions. So in purple, you see the metaphorical gland of this ant. I think this is Provenina. And the orange is the second peristernal muscle, just surrounding stuff orientation. And when you remove the gland, oh, there we go. See this little orange thing? This is the very reduced and very vestigial podiospherical muscle. It has been completely enveloped by the gland. And so prior to the advent of micro CT and the ability to look through things without tearing it apart, I think people just scoop the gland out, looking for the muscle. It's not there. I don't know what to do. And you see it here, right? This is a different group of ants, and they have a metaphorical gland more similar to pelicinids. And again, you see the little tiny red. So this is, I cannot take credit for the discovery of this. This is all Adrian. But you wouldn't have looked if I didn't tell him about the gland. So I take part in credit. Right, so this was cool because then we had something to compare the gland to, right? And I mentioned it's way easier to find function and sort of the evolutionary drivers of structure comparison. Well, I mean, that's what I said, what I thought. So we did some GC on the gland extracts of pelicinids, and we got 8 reasonable peaks. There's 5 here. The other 3 had incredibly long names that I don't know how to say, so I put in the talk. So the 3 that are in green are all volatiles known from metaphorical gland ants. We have hexadecanoic acid, linoleic acid. So, and metaphorical gland cocktail, it's basically a weak acid cocktail, right? That they groom onto their bodies and onto the bodies of their nestmates to kind of reduce microbial growth and retard microbial sort of getting in. There's also 2 other chemicals here, the henacosane and Z12-pentacosine. These were only previously known from scarab larvae. And I will fully admit that when I first got this GCMS report and I had looked at these, I said, oh, it's contamination. And I just put them out of my mind. But they will come back. So, we had some of these similarities in the gland extracts. Is this homology or this homoplasy? And so it's important, I guess I should mention sort of what functions this in ants. So this gland in ants is believed to be a prophylactic agent, right? It's really important in the social response. So this weak acid, as I mentioned, they groom over themselves, their nest mates, and sort of validated by the fact that it's mostly vestigial and arboreal ants. And so the selective pressure seems to be living in soil and being around soil-borne microbes and having to deal with that. Pelicinids obviously dig around in the soil. But the problem is, we couldn't find anything in any other. It's just these. And you have ants, which are the embodiment of sociality, right? And they have this gland, which is a key innovation in their social existence and being. And you have another group that is the very embodiment of solitary, right? They don't even need to mate, right? They don't even have that interaction. It's nothing. So what's the overlying pressure? And I kind of went back and forth with it for a while, and entomologists, basic, so plastic that shift and come and go. I personally think that this is a really, really deep level of convergence. I think, there's probably an underlying gene regulatory network within Hymenoptera, that there are hot spots around the body that glands are one of them produced in. Most glands are around sort of conjunctival joints, right? In the mandibles, stuff, places where it's easy, not the less direct work through, or in places where it's biomechanically easy to reach your legs and room with. Or in the case of, this area of the metapleuron, or the, eucoiline scutellar disc, right? There's not a lot going on underneath it. Not a lot of muscles around here. And internal real estate's at a premium in insects. So I think that might be a case, there's space to develop out this large gland. But I think these are probably convergent. And I should, mention that these aren't the only two Hymenopterans that hang out in soil, right? The pressure of soil-borne microbes is not exclusive to ants and pelicinids. You have specids, crabberonids, even Polistes. All of these have a immune system gland, right? In crabberonids and specids, you have a really cool gland on the antennae that basically it's a urine chamber for yeast. That's a symbiotic microbe, and they will coat their brood chambers with it, with the dirt, and it's a yeast that basically protects the egg and the developing larvae from nastier soil microbes. Polistes, paper wasps, they have antimicrobial peptides in their venom gland, and it has a similar function, right? They coat their brood chambers with it, and it kind of retards microbial growth. And so, you have these various forms of handling the same problem, but the fact that it occurred the same way in ants and pelicinids is still very strange. And so we talk about convergence versus homologous or function, right? Function is often easier to gauge from homoplasy because your selective pressures are the same. So you can get rid of everything else and just focus on that. So pelicinids, there's been behavioral studies, and they will stick their very, very, very long abdomen underground, sort of dig around and burrow. They actually have a very short ovipositor. Pelicinids, they have this fully telescoped articulating abdomen so that they can snake and they can dig away at soil looking for maybe the larvae. And what happens is after they dig around for a little while, they pull it out, they room it meticulously with this extract, go back in, do it again. And you have this pressure of close association with soil, similar to ants. So it's entirely possible that it has the same prophylactic function. But much like Bruce, I have a crazier functional hypothesis, and I cannot take full credit for it. I have to give some credit to the brilliant minds of Ted Schultz and Jeffrey Chicago. So I mentioned that there was those two compounds that are also known as sclerophyllite, which I would think are. The hypothesis is they coat their abdomens in this extract, and when they're probing around the ground, basically masks their probing. So that a Phylophaga beetle larvae is laying there, and this probe comes down, goes, oh, time to smell like another beetle larvae, whatever. And they won't kind of burrow away until it's too late to get stopped. Don't know, but it sounds good. Yeah, and so as was mentioned at the beginning, my real job is I'm a taxonomist. And I'm here working on Gibrionids, conifer sawflies. And fun fact, they have a pair interturbo gland that we don't know the function of, and it is very, very similar to the Day's organ of pumpkins, which we also don't know the function of. And so we're gonna have a quick recess and then another 30 minutes to talk. I'm done. That was it. Thank you for listening, everybody. Okay, stop sharing.
Replay post.
Thank you for that. Does anybody out in Zoom land have any questions? Or from the peanut gallery here?
Yeah.
I have a question or two or three. No, I'm kidding.
Go for it.
So did you find those glands only the females, or did you have the chance to look at
The pellucidant glands.
Yeah.
So for people in Zoom land who didn't get the question, she asked if the gland is only found in females. No, it's also in males. And this was actually something that was debated on the homology of the gland, right? So in ants, it's pretty much it's dimorphic. It's really only in the females, except in the really early ant lineages. You will find it in males. And so one of the early arguments for homology was, well, it's in basal lineages of ants, it's in both. Pellucidants, it's in both. Pellucidants are really older than ants. But this was an argument that was made. But no, it is in both, which is interesting, right? Because why do males need it if it's for like homoplasy?
Yeah, I was gonna ask you, what do you think that is going on with the males?
I mean, I think it's probably more efficient just to like have it in everything, right? Dimorphism is kind of something that there needs to be a pressure on to cause the dimorphism. But I don't know, mostly because it's really hard to get males because they all live in like Bolivia.
Yeah, and those compounds that were found that are I guess shared with scarab beetle larvae. What are the function of those compounds?
I think it was the paper I read. Basically, they just put a scarab larvae in a tube with a bee and shook it up and then ran it through a GC. Just like, here's all the stuff we found. It was pretty vague in the paper that it came from. I don't think they know the function in that either.
Was it on the outside of the larva or?
Yeah, I mean, in all honesty, I think they swabbed the outside of it to look at it as anything coming off. Representing its gland, and that was exuding something.
Do you think it's also like a prophylactic type thing?
It could be. I mean, it's most soil dwelling insects, especially larvae, have some prophylactic excretion, whether it's given to them by the parent when they are laid, or they exude it themselves. Termites have it, beetles have it, bees have it. Yeah, it's a pretty common. I don't know if that's the function. Most prophylactic excretions tend to be a really simple weak acid cocktail because you don't need much to prevent fungal growth. It just needs to be not available. Yeah, but I don't know. Yeah. Right.
I thought it was. I'm really interested to hear about the first section where you're comparing different species with the same gland.
Yeah.
Are there like biological differences between those species, or why? Yeah, why do you think it would be so necessary that you would have species-specific morphological differences with this
Right.
gland?
Yeah, no, that's a good question. Because the point of a gland, right, is it's the chemical that's being secreted is what's important, right? The morphology of the gland is kind of a redundant secondary thing. Yeah, I'm not sure. I don't really know. We don't know almost anything about the biology of cephalids. Mostly, they're virtually impossible to rear because they're usually, not even primary hyperparasitoids, but secondary hyperparasitoids of like reconids. So you have to maintain like 3 layers of biology. But yeah, I mean, it is. someone asked previously, like, oh, do you think all cryptic species complexes have something like this that just needs, a little puzzle piece that's just perfect, morphological barcode to identify it? I don't think so. I think I got incredibly lucky in working with a weird group that had some awful thing. But yeah, I genuinely have no idea why there's such a species level morphological variation. It doesn't seem necessary. Well, what would the selective pressure be?
And do they only have one gland each, or are they like?
do. So they have, that's awesome. So they have one gland, except the sister Seraphonidae, which is sort of the transition from Megaspilids, which don't have a water smooth evaporatorium. Transitionary group, which has a mixture of characters between them. The only one that has paired. So it's got two. And then in Masner, which is sort of the next most basal group within Seraphonids, it's basically one big one, but you can argue it's basically those two just kind of together. And you get up and it gets smaller and smaller and more close to the margin, and it slowly is modifying everything around it. Yeah, the earliest form of it is finally.
Yeah. What's with the term evaporatorium? Is that just an informal term, or does that have any?
That yeah, evaporatorium is a term used for the particulate cell structure of stink bug scent glands, right? It's an evaporatorium. It's basically thought that this reticulated sort of structure, right? In theirs, it's got like mushroom bodies, much more elaborate in stink bugs, but it's thought to work on the sprinkler system, right? Where like it hits sprinklers. But that's where the term evaporatorium came from. I didn't come. Yeah, let's hear it.
The Seraphonid stuff is super cool. I was thinking about Seraphonids this morning, and you answered some really nice questions there. But Pelosinids, have you done any histology on them, manual histology? Is there, so I'm just going to throw out also, is there anything like the sieve plate that's observed in ants?
No.
Nothing like
Yeah,
that.
so the pelicinid metapleural gland is very, very primitive form. There's no sieve plate, it's a single chamber. It's not like the two chambered internal ones you get in ants. One gland, it passively excretes out, right? There's nothing basically allowing any flexion of the pressure on it to cause an active secretion. The atrium has no bulla covering, right? The sedal row on the edge is really basic. You have ductual conglomeration, which you don't get in ants. Yeah, it's not anything as advanced. So I think if, ants definitely took it and ran with it because it had such high utility in being a social insect. And I think it just went super advanced relative to pelicinids that I think it's just does the job, whatever. So.
Well, if there's no questions out in Zoom land, I'll make a motion to close this meeting. Thank you.
One
Of
more
course.
question.
Oh, sorry, premature.
One more question, which is, in ants, we often think of the metapleural gland as kind of just leaking its secretion into the atrium and then spreading out onto the metapleural region in special areas, what have you. Now, is there anything at all on the legs, associated with the legs at all, or with the abdominal tip that are grooming structures? Because
Like a cleaning apparatus. I actually no. I wonder. I mean, I know they have pelicinids have basically grooming apparati on, but I think it's before that, which wouldn't really reach. I have to check. I don't know. I don't think on the hindlegs they have anything particularly unique.
just trying to imagine some way of contacting or, like soaking up the
Yeah.
secretion to put it onto something.
Yeah.
But doesn't seem to be.
No, I mean, so Andy Bennett at CMC was one of the behavioral studies. And so he had a couple in captivity and had some pilothaga. It was a fish tank basically with dirt, pilothaga beetles in there, through Pelsininic. They're not very good at finding hosts. They're just literally digging blind and just feeling around in the soil. But they would do this for a little bit, and they pull it up, and they would take their hind legs, and they would kind of rub it against their podial region, and then groom the abdomen completely, and then go back in. So, I mean, yeah, they have to have some way of transferring it. But I don't know of any like, very specific grooming structure that you would find in this motherfucker that doesn't have. A lot of unknowns. Everyone should study glands.
All right, now I would like to make a motion to close this meeting.
Seconded.
Thank you. But of course, if anyone has any more questions, please ask our lovely guests.